At the start of the project, there wasn't a cattle genome. Things like the array technology was still relatively in its infancy, so the range of tools we had at our disposal was rather limited, and the amount of data that was generated was not completely overwhelming. So there was a realistic chance that the sort of tools that we had routinely available at that time would be able to handle the data reasonably well. However, as the project has continued, we've been right in the middle of the genomics revolution. So year on year, the biology technology has leapt ahead. The amount of data that it is possible to get your hands on each year has grown and grown. We have gone from the situation where we had no cattle genome to a rather poorly constructed cattle genome to a really quite good cattle genome to we now have the genomes of many different cattle, including specifically the types of African cattle that we've been interested in. So from having no cattle genomic data at the start of the project, we now have about 10 billion bases of cattle data to work with. Now, in some ways, that's incredibly exciting. It means suddenly we have the information and the tools to really begin to understand this problem, to stand a realistic chance of actually cracking it. On the other hand, how the hell do you deal with 10 billion bases of cow genome? That's a lot of data, and that's not the sort of thing that we had the tools or the infrastructure to deal with. In the same way that from thinking about we might be able to get a little bit of information on the genes expressed, we now have data on gene expression from multiple tissues, all the genes expressed in multiple tissues across around 100 cattle. And that's fun because that's a lot of data. And if that's all there was, then that would be straightforward. But at the same time, our biological understanding has massively increased. So the biological framework in which we have to put this knowledge has got itself significantly more complicated. We understand a lot more about gene networks, gene pathways, the way the immune system functions, the way that cholesterol metabolism works, the way that lipid metabolism works. And if we're going to properly make use of the data, we have to take these very large, complicated data sets and understand them in the context of current biological knowledge. And that suddenly wasn't quite what we could do with the existing compute resources. That was an interesting challenge. And that is what has then driven a lot of the way we had to move the project, because it was clear that as bioinformaticians, we know a little bit about how to work with data. We understand a little bit about computer infrastructure, but we do not have the skill sets to work with those large and complicated data sets. That if we wanted to make progress in this area, we had to very much expand the sorts of collaborations we were involved with, which meant bringing in a really strong e-science, computer science group. The length of time it takes you to develop good computer infrastructure is longer than the timescale at which the biology data is changing. So we're in a constant Red Queen's race. We can never sit down and think, what's the most beautiful system we could develop? Anything we develop has to be easily changed. It has to be rapidly modifiable. So it has actually been very difficult to actually get across the idea to, say, some of the IT team that what we need is that flexibility and scalability as much as we need a beautiful piece of engineering for one specific problem. So it's been getting that flexibility built in and actually getting the communication going so that those messages could go backwards and forwards properly has been one of the more challenging parts.